In this screencast, we're going to look at the chemistry of carbon, and really what I mean by that are the building blocks of carbon molecules and the macromolecules that are carbon-based. Why study carbon? We talked about why study water. It's just so common in organic molecules. The definition of organic molecules are carbon-containing molecules, and big percentage of water and a huge percentage of what's left over are carbon compounds. And these are the big four nutrients or carbon compounds that are organic: carbohydrates, lipids, proteins, nucleic acids. Hydrogen and oxygen are probably the most abundant elements in nature, and carbon is next. So organic chemistry is the study really of carbon compounds. And things that you'll just know out of experience is that carbon has an atomic number of 6. It's got 6 protons, meaning it's also going to have 6 electrons. Now, what does that mean? That means that there are 2 electrons in the first energy shell or level, and then 4 in the outer valence shell. What is that important for? Why is that important? It's because that would leave 4 bonds that could be made. And 4 bonds could easily mean 4 hydrogen atoms to fulfill the octet rule. So these organic molecules are put together like the old Tinker Toys, if you remember those. The carbon is going to have, again, spaces or places for covalent bonding to take place. And they are very structural and stable. So for instance, sugars like glucose, C6H12O6, or the methane or the ethane here. The carbon building block with hydrogens on the outside, but they can be arranged in different ways. We've already talked about hydrocarbons and the properties of those, but again, they're important to recognize. They are organic molecules, right? So we're going to deal with them a lot in biology. Hydrocarbons, if you remember from our lab, they form non-polar, therefore non-soluble or insoluble in water solutions. So hydrocarbons are nonpolar, they're not soluble in water, they are hydrophobic. All of those words you need to be able to use and kind of interchange and talk about their relationship together. So hydrocarbons, they're very stable. They're very stable. They're not going to dissolve in water. They're not going to dissociate and break apart. There's very little attraction between them and other molecules. They're just good foundational structural molecules. As we already stated, hydrocarbons can grow just by adding carbons. And of course, if you're going to add carbons, the number of hydrogens are also going to grow just to fulfill each individual atom's octet rule. But just adding the carbons is going to change greatly the property of the molecule. And if you look here, here we have butane, which is a 4-carbon molecule, right? Isobutane, well, it's also a 4-carbon molecule. It's just that there's actually branching here. So you can save memorization and recognition of all of these for your organic chemistry class someday. But just to show you that it's not just about the numbers of, for instance, carbons, it's also how they're arranged. And so different molecules with the same molecular formula, in other words, the same number of carbons, same number of hydrogens, etc., just with different shapes, those are called isomers. And isomers, they can actually have quite different functions. And actually isomers are a lot of times ineffective as medicines, because even though the formula, kind of the ingredients are the same, the outcome can be way different just because of the way that the shape is going to determine its biological properties. This brings up quite a bit of diversity between hydrocarbon and organic molecules. That if you substitute other atoms other than hydrogen basically around the carbon, you can drastically change the effects. So here, just as an example, you've got ethane versus ethanol. So in ethanol, the hydrogen, one of the hydrogens is actually replaced by a hydroxyl group. Remember that as a functional group. And what do you remember about the important interaction between oxygen and hydrogen? Think electronegativity. So which is polar and which is nonpolar? And if you think about the polar structure of the OH bond in water, remember that oxygen is a little bit more, relatively quite a bit, electronegative. So than hydrogen, it's going to win that little electron tug of war. So that's going to cause polarity. So an ethanol molecule, just because of the addition of the OH group, is going to become polar. Well, what does that mean? What are the biological effects of that? Is it going to be hydrophobic then or hydrophilic? Well, if it's polar, it's going to be soluble in water. Therefore, it's going to be hydrophilic. So this brings us to a little more discussion about functional groups. You should already be able to just simply recognize them. And it's important because you want to know that the structure is going to determine the function. So a little bit more explanation on functional groups. These are just parts of organic molecules that are involved in the chemical reactions. They're attached to what are called R groups, which are essentially for now just the representative group, the kind of the rest of the molecule, the base of the molecule. But you add these functional groups and it will determine how they react with other organisms, how chemistry is actually going to take place, how reactivity is going to take place. So these are really the big six. There's a couple of different examples within those, but we want to be able to identify these: a hydroxyl, a carbonyl, a carboxyl, amino, sulfhydryl, or a phosphate. And it's going to affect their reactivity. For an example, the male and female hormones, testosterone and essentially estrogen, but a derivative of this, they differ from each other only by the attachment of a different functional group to the identical original skeleton. That's all that makes the difference. Essentially a hydroxyl versus just an oxygen attached to the rest of the group. So, as you probably know, the actual function that protein, that hormone is going to play out in a body be quite a bit different. Already touched upon hydroxyls. Really, with those hydroxyls, those are in a class called alcohols. They have that typical ending of the ol or the ol, like ethanol. A hydroxyl again is going to make the compound polar. Again, why? Because of the electronegativity of the OH group, meaning that this is going to become a polar part. Therefore, it's going to have something to do with its affinity for or against water. In this case, polar, like dissolves like, it's going to be soluble in water. So things like methanol, ethanol, propanol, butanol, all of these gases, or actually these are alcohols that are derivatives of gases such as methane, ethane, propane, butane. Carbonyls, this you look for that distinctive C double bond O. So you've got an oxygen double bonded to carbon. And these things, these carbonyls, there's a couple different types depending on where in the molecule they exist. But carbonyls, you might hear things like formaldehyde, even one called cinnamaldehyde, which gives some cinnamon flavor in perfumes. Something like acetone, it's a common ketone that's used as a solvent for things, like fingernail polish remover or to remove paint strippers or things like that. So these, a carboxyl can be a little hard to differentiate between the carbonyl and this, and actually this contains, a carboxyl contains a carbonyl group. But the distinctive COOH structure, a carbon double bonded to oxygen, and then in addition also bonded would be a hydroxyl group, or an OH group. So compounds with these are typically going to be acidic, and including fatty acids, amino acids, as part of organic group. We notice this group, NH2, we notice that that's a part of an amino acid. Those are the amine groups. And not to really contradict here, but we really want to just recognize that that is an amino group. You will find this, NH2 group, in every amino acid. A sulfhydryl, here you've got the distinctive sulfur there bonded to a hydrogen. They form a class called thiols, and these groups stabilize the structure of proteins. They are often involved or responsible for a number of smells, as weird as that sounds. Those thiols are responsible for like skunk smells, and a particular derivative of a thiol is added to natural gas, so it has that sulfury smell to indicate leaks. Grapefruits and citrus fruits, but grapefruit kind of has that distinctive smell as well. Maybe not bad, but bad breath, onions, garlics, a lot of these things have thiols as a part of the macromolecule. And thiols also, they readily bond to proteins, especially in the skin. That's why, I mean, you handle onions and or garlic, or even eat those things, the smells can linger because they attach onto the resident proteins that are already there. The last of the functional groups that you need to know, pretty noticeable, a phosphate group, just essentially a phosphorus atom that is bound to 4 different oxygens, one with a double bond, but the R group would attach to one of the oxygens. They have kind of a big negative charge, which has to do with their reactivity. Phosphates are a part of ATP, adenosine triphosphate. That is the molecule we'll deal tons with down the road. The molecule of chemical energy for living things. They're a component of DNA, of nucleic acids. They're a part of every nucleotide. They are a part of many enzymes involved in reactions such as photosynthesis and respiration. They're a part of phospholipids that you've learned about, hydrophilic heads in phospholipids. So they're a big component of cell membranes. So there's a lot of energy available here. When you break this big molecule off of an R group, of a big macromolecule, a lot of energy can be released, energy that can be harvested to do work. So where we'll go from here is we're going to talk about the macromolecules, as I mentioned already, carbohydrates, lipids, proteins, and nucleic acids. We'll look for certain functional groups and how they are a part of the macromolecules, because, again, that will help us kind of pinpoint how they react and how they behave in organic molecules and in cells, etc.